Hello everyone. So today let us start with Unit 5 of Semester 3, that is this excretion. So in the excretion we are covering structure of nephron, mechanism of urine formation and counter current mechanism. So let us start with the structure of nephron. Nephron, as you already know, that it is the basic structural and functional unit of kidney. It is mainly composed of a renal corpuscle and a renal tubule. So renal corpuscle is the Bowman's capsule and glomerulus part, and the tubule is the proximal convoluted tubule, then the U-shaped loop of Henle, and then the distal convoluted tubule. So PCT, Henle's loop, and DCT, then together they are forming this collecting tubule. It is the DCT, they are of all the nephrons are going there and getting merged with the collecting tubule. So this is the structure of kidney from the inside. These are called renal pyramids, and the outer portion is called the cortex and the inner portion is called the medulla. So based on this portion of the kidney and the location of the nephrons inside the kidney, there are two types of nephrons. One is the cortical nephron. They are present within the renal cortex. So 80% of the total nephrons are cortical nephrons, and they are of course shorter in length. But the medullary nephrons, juxtamedullary nephrons, they are actually having long loops of Henle, and this loop of Henle, they dive right into the renal medulla, and so they are about 20% of the total nephrons. So this we already know actually, that the afferent arteriole from the renal artery, it carries the oxygenated blood and it enters into the glomerulus, and the deoxygenated blood, that is the efferent arteriole, the oxygenated blood only from the efferent arteriole comes out after being filtered by the Bowman's capsule, and it goes through the capillary network that is there, which is known as peritubular capillaries, which are surrounding the actually renal tubule. So this then comes out in the form of renal vein, which carries oxygen depleted blood. So this is the basic structure of the nephron, and according to the functions, like which part of the nephron is responsible for what, here as you can see that the glomerulus is of course responsible for filtration, the PCT is actually responsible for reabsorption, and the descending loop of Henle is for aquaporins, and we allow the water to pass from the filtrate, and the ascending loop of Henle is for reabsorption sodium and chloride ions, and then the DCT for selectively reabsorbing certain solutes and also secretion, and collecting duct is of course reabsorption of water. So these are the basic functions. We will go into further details about that in the next slides. So this is the renal corpuscle. Just this portion has been zoomed out here, and it is given like this. So this is the inside of the renal corpuscle. So the Bowman's capsule, which is the one end of the blind end of the nephron, it is a double-walled cup-shaped structure. This is known as Bowman's capsule or the renal corpuscle. So this capsule is enclosing a tuft of capillaries or a bunch of capillaries, which is known as the glomerulus. So this glomerulus and Bowman's capsule, they together it's called the renal corpuscle. So this capsule is having three different layers basically. The outer parietal layer, which is composed of squamous epithelium, this blue color thin layer. The inner, there is a basement membrane, like this is the basement membrane, as you can see that the basement membrane is there, and also there is an inner visceral layer, the light blue color, that this is composed of large nucleated cells. These cells are called the podocytes, and they bear sharp finger-like projections. These are known as pedicels. So these finger-like projections, they wrap around the glomerular capillaries, like these are the capillaries, as you can see. So these podocytes and pedicels are wrapping around the glomerular capillary surfaces. So these three layers, they are actually coming from the Bowman's capsule, and the space between the visceral and the parietal layer, this space is known as the Bowman space or the capsular space. And the glomerulus, it arises from the afferent arteriole, and it ends with the efferent arteriole. So this there is a diameter difference between these two arterioles, and therefore the pressure difference is there. Like the blood is in high pressure in these capillaries. So therefore this pressure difference is causing the filtration to happen. So this we will go into further details. Now as you can see that there are three different colored cells over here. This is the So first let me tell you why the DCT is shown here. So in one nephron, if you can see, when the Bowman's capsule, the PCT is here in continuation, there is a loop of Henle, and this is the DCT. So when the DCT is going up, it comes in close contact with the renal corpuscle. So here the DCT and the arterioles, they are close together like this. So this is given here. So if this is the DCT, like it's a cross section of the DCT, then these cells are known as the macula densa cells. And these cells, green colored cells of the afferent arteriole, these are the granular cells or the juxtaglomerular cells. And these cells are the violet colored cells, are basically forming like a matrix kind of thing. These are stromal cells. These are extraglomerular cells. That means they are not part of the glomerulus. So these three kinds of cells, they together form the juxtaglomerular apparatus or the JGA. Like coming to that in detail. So the JGA is basically this apparatus, comprising of three different cells: the juxtaglomerular cells or the granular cells, the macula densa, and the mesangial cells. So this is located between the afferent arteriole and the returning DCT of the same nephron, like I have already told you. So it is responsible for both intrarenal mechanisms and extrarenal mechanisms. Now what is intrarenal mechanism? It is the tubuloglomerular feedback. So what happens is that it is an adaptive mechanism. So basically the blood is coming from here, it is getting filtered over here in the glomerulus. Now how much is the concentration of the salt concentration of the here in the macula densa, that actually triggers that how this fluid pressure or filtration will be carried out in the Bowman's capsule. Because in the DCT, that fluid is already, the urine is formed over here. So if the salt concentration is very much high here, then it will trigger the afferent arteriole cells, the juxtaglomerular cells, to secrete renin, which in turn will cause the GFR, that is the glomerular filtration rate, to go down. So if there is a high salt concentration here, obviously it will trigger the cells over here. So to slow down the filtration rate, to slow down the filtration process, so that the urine is formed properly. And of course over here, this is an auto-regulatory mechanism. So this is a feedback kind of mechanism. The renal tubule is giving feedback to the glomerulus that how to control the glomerular filtration rate. So how this is done? This is done when the juxtaglomerular cells are getting a trigger, specific like signal from the DCT, that is the macula densa, that we have a very strong salt concentration. So here in response to specific stimuli like this, like low blood flow is there, or decreased sodium chloride is there. So these are specific stimuli which triggers the juxtaglomerular cells to synthesize and store renin. So renin is an enzyme, and it is considered as the effector arm of the renin-angiotensin-aldosterone axis. So this is the extrarenal mechanism that I was talking about. This is a one kind of an axis that is happening over here. I will go into the details. So what happens is that the JGA, the juxtaglomerular cells, they are secreting renin. So macula densa, it is the region of the DCT. Now it can be considered in this region, the epithelial cells are more closely packed than the other regions, as you can see over here. So this is the sensory arm of the renin-angiotensin-aldosterone axis. They sense the decreased sodium chloride delivery, which determines downstream function. So they are also involved in the mechanism of tubuloglomerular feedback, like I have already said. Now they sense that whatever constant salt concentration is there, and they simply relay this message to the juxtaglomerular cells whether to secrete renin or not. Now the mesangial cells, they are basically there, they are forming connections via actin and microtubules, and they allow selective vasoconstriction and selective vasodilation. That means constriction of the blood vessels and dilation of the blood vessels of the renal, afferent and efferent arterioles. So they basically control the vasodilation and vasoconstriction of the arterioles, and therefore they control the blood flow into the glomerulus. So how is this renin, angiotensin, aldosterone system, or the RAS system that we in short say it as RAS, this is how helping our filtration in the kidney. So renin is actually enzymatically converting angiotensinogen into angiotensin 1, and then its release is stimulated by paracrine signals from the JGA in response to decreased extracellular fluid volume. So here, macula densa, it senses low fluid flow or low sodium concentration. So when there is a low sodium concentration, it is signaling the juxtaglomerular cells to secrete renin. Okay, now kidney releases this enzyme renin into the blood. So liver also releases angiotensinogen into blood. It produces angiotensinogen and is releasing it into the blood. So in the blood we have angiotensinogen and we have renin now. So renin will convert this angiotensinogen into angiotensin 1. Now angiotensin 1 will be converted via angiotensin converting enzyme ACE in pulmonary blood because it is coming from the lungs, and it will be converted into angiotensin 2. Now what will happen, this angiotensin 2 is a potent vasoconstrictor, so it will promote widespread vasoconstriction overall. So obviously it will regulate the blood pressure. Now vasoconstriction as well as constriction of the efferent and afferent arterioles of the glomerulus. Now what will happen is that the efferent arteriole will be constricted more strongly than the afferent arteriole. So therefore increasing the GFR. So therefore more blood will go inside the glomerulus, but less amount of blood will be coming out from the glomerulus. So the filtration rate will obviously increase because the blood will be present in the glomerulus for a longer period of time, and therefore it will get filtered more more extensively. So this will okay, so now this angiotensin 2 has done its vasoconstriction activity or whatever. So in instances of blood loss or dehydration, this angiotensin 2 reduces both GFR and renal blood flow, therefore limiting the fluid loss and preserving the blood volume. So when there is a blood loss in the body, or when we are going through a period of dehydration, then this angiotensin 2 actually acts upon the blood vessels and it reduces the blood flow in both afferent and efferent arteriole. Therefore what happens is that there will be less amount of fluid in the glomerulus, therefore less urine will be produced and less water volume will be lost from there. So its release is actually stimulated by a decrease in blood pressure, and so the preservation of adequate blood pressure is its primary role. So what happens to the angiotensin 2 then? So angiotensin 2 is obviously in one channel it has gone to vasoconstriction, and in the other way it has gone to adrenal cortex to secrete aldosterone. So why aldosterone is secreted? Now aldosterone stimulates the sodium uptake on the apical cell membrane in the distal convoluted tubule and collecting ducts. So now aldosterone will trigger the DCT to uptake sodium, and in the collecting ducts as well as in the DCT to uptake sodium. Now what will happen is that when there will be an uptake of sodium, so therefore the low sodium concentration will be somewhat replenished. Now this DCT also has this enzyme called ADH, antidiuretic hormone. This hormone causes the aquaporins to move to the collecting duct plasma membrane, which increases water reabsorption. So now the water will also be reabsorbed, and therefore the loss in the volume, fluid volume in the DCT will also be replenished. So in this way the renin, angiotensin, aldosterone system is working. So aldosterone is called the salt retaining hormone. Why? Because it is helping in the uptake of sodium, sodium reabsorption in the nephron, in the DCT. Here. So angiotensin 2 has gone to the vessels for vasoconstriction. Now it has gone to adrenal gland and it is causing the secretion of aldosterone. Aldosterone is now acting upon the kidney and it helping in the reabsorption of sodium chloride and water. So now this It promotes sodium reabsorption and promoting the retention of water. It is also important in regulating potassium and promoting its excretion. So sodium will be uptaken and K+ will be released. So this dual effect on two minerals and its origin in the adrenal cortex is therefore explains its designation as a mineralocorticoid. So therefore the aldosterone is also known as a mineralocorticoid. Now this renin has an immediate effect on blood pressure. Therefore, due to this angiotensin II stimulated vasoconstriction and a prolonged effect through sodium recovery due to aldosterone. So at the same time that aldosterone causes increased recovery of sodium, it also causes greater loss of potassium. So obviously in one way sodium will go inside and potassium will go outside. Now antidiuretic hormone or ADH, this is very important hormone in urine formation. So it basically promotes the recovery of water from the filtrate, from the fluid volume of the renal tubule, because when there is a loss of water inside the body or the body is dehydrated, then preserving water is very important. So what happens is that it helps in the retention of water, preserving water from the urine, and it decreases the urine volume. So how it does that? It does by stimulating the movement of aquaporin proteins. So there are certain proteins called aquaporin. They stimulate those proteins to go inside the cell membrane, and these proteins can form the water channels. So it will allow the water to flow from one side to other side, and therefore the water will be taken back from the tubule and it will go inside the blood volume again. So from there it enters the vasa recta capillaries, like I said, and it returns to circulation. So water is attracted by high osmotic environment of the deep kidney medulla. So it is very much hyperosmotic. So water will always move from hypoosmotic to hyperosmotic region, and therefore in the kidney medulla it is hyperosmotic. Therefore it is coming out from the renal tubule into the kidney capillaries. So this process allows for recovery of large amounts of water from the filtrate back into the blood. So what happens when ADH is absent? Then aquaporin channels are not formed. Therefore huge amount of water is excreted in the form of dilute urine, and therefore there will be a huge loss of water from our body. Now the function of aquaporins is to allow the movement of water across the lipid-rich hydrophobic cell membrane. Okay. So coming to this renal tubule part, this renal tubule is basically a long structure that it consists of three parts. The first is the PCT. Because of its proximity to the glomerulus, it is called the proximal convoluted tubule. This part usually stays in the renal cortex. The second is the loop of Henle. It obviously goes into the renal medulla. This is the outer medulla, this is the inner medulla, and the third part is the DCT or the distal convoluted tubule. It is also coming back again in the renal cortex. So this DCT is the last part of the nephron, and it connects and empties its contents into the collecting duct. So here you can see this is the cortical collecting duct. As it goes into the medulla, this is the medullary collecting duct, and all these collecting ducts, they collect the contents from all the nephrons, surrounding nephrons, and if fused together they enter the papilla of the renal medulla. So here it goes and dumps its contents over here. So what is the function of this renal tubule? It is the main function is reabsorption, and this process can either be through active transport or passive transport. So in addition, secretions by the renal tubule help in urine formation without affecting the electrolyte balance of the body. So basically reabsorption, secretion is very important, and therefore this renal tubule is playing a great role in urine formation.